My name is Wu Zhang, and my report topic is real-time PCR test. I will introduce what is real-time RT-PCR, how does real-time RT-PCR work, and why use real-time RT-PCR. As of May 5th, the respiratory illness caused by 2019 novel coronavirus has resulted in 68,279 death cases in America. Coronavirus are a group of viruses that can cause upper and lower respiratory tract illness and have a characteristic spiky appearance that resembles the SARS corona. Although many common coronaviruses usually cause mild to moderate illness, some can cause severe illness that can progress to an fatal pneumonia, which occurred during the MERS coronavirus and SARS coronavirus outbreak of 2015 and 2003, respectively. Since there is a 2% death rate from 2019 novel coronavirus, and it may be transmitted by exposed individuals who show no symptoms, effectively screening tests for the virus are vitally important to controlling this outbreak. CDC has developed a new laboratory test kit for testing patient specimens for severe acute respiratory syndrome. The test kit is called the Centers for Disease Control and Prevention 2019 Novel Coronavirus Real-Time Reverse Transcriptase Diagnostic Panel. This test is intended for upper and lower respiratory specimens. So, what is real-time RT-PCR? Real-time RT-PCR is a real-time reverse transcriptase polymerase chain reaction in which application is actively monitored as the reaction progresses. Real-time RT-PCR is now the most widely used method for detecting coronavirus. How does real-time RT-PCR work with the coronavirus? The first step is swab. A test for 2019 coronavirus starts with a sample. Usually, this is done with a swab that goes deep in a person's nose and throat. This is then placed into a special transport media and sent to a testing laboratory. Lysis buffer. Lab scientists combine small amounts of a person's sample with a lysis buffer that remove substances such as proteins and fats and extract only the RNA present in the sample. This extracted RNA is a mix of the person's own genetic material and, if present, the coronavirus RNA. RT-PCR machine. During RT-PCR, the RNA is reverse transcribed to produce a matching pieces of DNA called a copy DNA. This copy DNA, in turn, serves a template for PCR to make enough copies to analyze. A standard real-time RT-PCR set time usually goes through 35 cycles, which means that by the end of the process, around 35 billion new copies of the sections of viral DNA are created from each strand of the virus present in the sample. Fluorescent probe. The special fluorescent probe for each gene in the coronavirus that the scientists want to target is added in the test. Each probe is on a mission to find its specific targeted gene. As new copies of the viral DNA sections are built, the marked labels attached to the DNA standards and then release a fluorescence dye, which is measured by the machine's computer and presented in real time on the screen. This is RT-PCR panel primer and probe sequences. Result: the machine records spikes and on the data graph that track the glowing as it gets brighter and brighter, revealing when a patient has a coronavirus 2019. Negative results won't show the spikes. The computer tracks the amount of the fluorescence in the sample after each cycle. When the amount goes over the certain level of the fluorescence, it confirms that the virus is present. Scientists also monitor how many cycles it takes to reach the level in order to estimate the severity of the infection. The lower the cycles, the more severe the viral infection is. Finally, control. Control samples included in RT-PCR and on the sample plate. These controls ensure that the real-time RT-PCR run performed as expected and was free of contaminants, and the amplified DNA is truly from SARS coronavirus. So why use real-time RT-PCR? The real-time RT-PCR technique is highly sensitive and specific, and can deliver a reliable diagnosis and as fast as 3 hours, though usually laboratories take on average between 6 to 8 hours, compared to other available virus isolation methods. Real-time RT-PCR is significantly faster and has a lower potential for contamination or errors, as the entire process can be done within a closed tube. It continues to be the most accurate method available for detection of the coronavirus. To detect past infections, which is important for understanding the development and spread of the virus, real-time RT-PCR cannot be used as the virus are only present in the body for the specific window of time. Other methods are necessary to detect, track, and study past infections, particularly those that may have developed and spread without symptoms. As real-time PCR testing continues in the coming days, we may see a rise in possible cases. But this is a progress and is an essential step in halting the spread of this disease, obviously. That's all. Thanks for the listening.